Welcome back. This is going to be our second screencast for Chapter 7. Now in this screencast we are going to focus on eukaryotic cell structure. Now if you remember back to 7.1 we had looked at two different types of cells. We had looked at a prokaryotic cell and we had looked at a eukaryotic cell. And I said it was really important that you make sure you know the difference between the two. If it's a prokaryotic cell, you're probably looking at a bacteria. If it's a eukaryotic cell, it's going to be any other cell out there besides a bacteria. Now if it's a prokaryotic cell, you're looking at a pretty small cell. So it's going to be pretty tiny. If you're looking at a eukaryotic cell, it's going to be relatively large in comparison. Now if it's prokaryotic, it's not going to have a nucleus, so no nucleus. It does have DNA, but there's no nucleus. If it's a eukaryotic cell, it definitely will have a nucleus. Now, prokaryotic cells are not complex, so they're actually considered pretty simple cells. Eukaryotic cell, on the other hand, is very complex, which means that it has lots and lots of cell parts, and that's what we're going to look at in 7.2. So looking at this first slide, you're going to notice we have a word in the upper left-hand corner, and this word is organelles. Now organelles are considered membrane-bound specialized cell parts, so they do specific things for the cell. Oftentimes they are described as little organs. Now if you think about our bodies, we have organs that are specialized to do certain things for us. If you think about the heart is there to pump blood to various parts of our body. If you think about the lungs, they're there to take in oxygen and of course release CO2. And so all of our organs are there for a reason. They do specific things to make sure that we stay healthy and basically survive. Same thing for the cell. The cell has structures called organelles, and again they're called little organs, and they are specifically there to make sure the cell gets what it needs to survive. Now all of these organelles are found within the cytoplasm of the cell. Now the cytoplasm is the fluid-filled space in the cell, so it's basically there to support and to nourish the different organelles. So the first organelle we're going to look at is going to be the nucleus. Now this is often considered the most important cell structure. Now the nucleus has two functions. The first function is to act as the control center or the brain of the cell. So its job is to basically tell the other cell parts what they need to do. And the second function is to use its coded instructions to make various proteins and any other molecule that is necessary within the cell. Now these coded instructions are going to be found in the form of DNA. Now the DNA is going to be packaged in one of two ways. It's either going to be packaged as chromosomes, which means that the cell is probably actively dividing, so it's going to split into two, so that DNA needs to be split evenly between two cells. And when you take that DNA and you compact it into chromosomes, it allows that split to occur rather easily. If the cell is not actively dividing, then it's probably going to be in the form of chromatin, which means the DNA is going to be scattered loosely throughout the nucleus. Now, as we said before, the nucleus is considered a membrane-bound sac, and you can see the nuclear membrane right here, and it's represented by this light purple line that you see in this cutout section of the nucleus. Now, the nucleus also has pores, which basically means it has super tiny holes in the membrane itself. And just like the cell membrane, those pores are there to let things in and to let things out. So within the nucleus, we need to make sure that we mention the dense dark region that's found within that nucleus. And that dense dark region is going to be called the nucleolus. Now the nucleolus is located right about here in the nucleus. Now this nucleolus is important because it's going to be there to produce ribosomes. Now those ribosomes become important because the ribosomes are going to be there to produce protein. And those ribosomes are going to be made primarily of RNA. But the only way the protein can be produced is if the ribosomes can actually find their way out of the nucleus. And the only way they're going to be able to do that is if they pass through the nuclear envelope. And the only way they pass through the nuclear envelope is if they go through what we call the nuclear pores. And we've mentioned those on the previous screen. So once the ribosomes are created, once they've been assembled, they're going to find their way out of the nucleus through these nuclear pores and actually find their way to what we call the endoplasmic reticulum, which is what you see right here. And that is going to be where protein is going to be produced within the cell. So the next organelle we're going to look at is going to be the mitochondrion. Now this particular cell part is really important to the cell because it acts as the power plant of the cell. Now what we need to think about here is all the food that we consume has to be changed over into a form that our cells can actually use. And so the mitochondrion is going to convert all the chemical energy that is found within the bonds, within the molecules of the food that we eat, into a form that can be used by the cell. And typically that form is going to be ATP. So that food energy needs to be converted into ATP so the cell can actually use it. Now over here on the right hand side you're going to notice we have 3 mitochondrion being represented within our cell. Now one thing I'm going to need you guys to be able to do is I need you to be able to recognize the difference between a mitochondrion and a chloroplast because they kind of look similar to each other. So what you need to look at here is you need to look at the wrinkled pattern within the mitochondrion. That's a dead giveaway that you're looking at a mitochondrion. If it's a chloroplast, it tends to be in the form of stacks. Sometimes they refer to them as sort of a stack of paper plates within the structure. So a chloroplast is often shaped similar to a mitochondrion, but the inside is going to be a little bit different. So it's really important that you guys recognize the difference between the two. So thinking back when we talked about the nucleus, we had mentioned something called the nucleolus. And remember the nucleolus is that dense dark region within the nucleus. Now the ribosomes are going to be produced within the nucleolus. Now remember I had mentioned that the ribosomes were there to build proteins. Now the only way that we can build proteins is by using a set of instructions that come from the DNA. Now again, proteins are considered a macromolecule, so it's a big molecule. It's considered a polymer, so it goes through polymerization. Now a polymer is built using smaller monomers. Those are the small units when they're put together produce the polymer. So since we're talking about proteins, remember amino acids are considered the monomers of proteins. So again, knowing which amino acids need to be put together, the only way we can figure that out is by using the instructions that are given by the DNA. And remember, the DNA is going to be found within the nucleus. Now, these ribosomes can be either free-floating, which means they're located within the cytoplasm itself, and you can see a ribosome here, you can see one located here. Or they could be attached to a structure called the endoplasmic reticulum, and that's going to be located right here. So all of these small dots that you see along this structure, those are ribosomes. Now remember, the ribosomes are made of RNA, and that RNA is going to be found within the nucleus. So during our discussion about ribosomes, we had mentioned there were 2 different places that you might find ribosomes. One of those was scattered throughout the cytoplasm, and the second was attached to another cell structure, and that cell structure was the endoplasmic reticulum. Now sometimes we call the endoplasmic reticulum simply ER. It's a little bit simpler to say ER than endoplasmic reticulum. Now there are 2 different types of ER. We have a smooth ER and we have a rough ER. Now on the right hand side here you can see surrounding the nucleus we have the rough ER on the right hand side, and again it's considered a rough ER because it has ribosomes attached to this structure. So you can see the ribosomes dotting the ER. If it's a smooth ER, you're going to notice we do not have any ribosomes that are attached to the ER. So you can see a difference between the two. Now if it's a smooth ER, its job is to build lipids, also known as fats, and its other job is to produce carbohydrates, again more commonly known as sugars. And those sugars could be simple or they could be complex. Now again, we have ribosomes, and the ribosomes are there to produce proteins. So if you notice, the ER is really important because it actually produces 3 out of the 4 macromolecules we had discussed back in 2.3. The next organelle we're going to look at is the Golgi apparatus. Now, some people refer to the apparatus as a Golgi body, and others refer to it as a Golgi complex. In my class, I would prefer that you use the terms Golgi apparatus. Now, the Golgi apparatus is going to take in protein that was produced from the ER. Now, more specifically, the rough ER. Now, remember, it's rough ER because it's dotted with those ribosomes, and the purpose of the ribosomes was to produce protein. Now, the Golgi apparatus is there to transport. It's there to move things from place to place within the cell, or even maybe transport things out of the cell. Over here on the right-hand side, you're going to notice the Golgi apparatus is located right here. And if you look really closely, you're going to notice these very small, tiny pockets that have been produced by the apparatus. Sometimes these sacs are called vesicles, and those vesicles contain the finished cell products, and in this case it would be protein. And again, that protein needs to be shipped to various parts of the cell or outside of the cell where it can be used. So similar to our own bodies, the cell also has special cell parts that are used to clean up the cell. These special cell parts are called lysosomes. Now, lysosomes perform two functions. One of them is to act as the stomach of the cell. So we have special sacs, and if you look over here on the right-hand side, you can see these sacs that are filled with digestive enzymes, just like our own stomach. And they are used to break down lipids, fats, carbs, which are sugars, and of course break down the proteins so they can be used by the cell. The second job of a lysosome is to digest or get rid of any worn out cell parts or organelles that are found within the cell. Just like any other part, they get wore out, they can no longer be used, and you have to get rid of them. And so the lysosomes are going to act to do just that. So the next cell part we're going to look at can be tricky in a couple of different ways. Now the reason they're tricky is because centrioles normally will only be found in animal cells. So if you're looking at a plant cell, you're probably not going to see centrioles. The second way it can be tricky is that they normally do not become visible unless the cell is actively dividing or going through mitosis. Now over here on the right hand side, you can see the centrioles being represented, and they're usually represented as a pair of bundled tubes, and those tubes are going to be made of protein. So one more time, this is an animal cell. They would be visible in this type of cell, but only if the cell is dividing. Now what's really hard for a lot of people to believe is that the cell actually has a skeleton. Now this skeleton is not made of bone like our skeleton, it's made of protein. Now this protein is going to come in the form of microtubules or microfilaments. Now both of these are made of protein. Now it's considered a cytoskeleton, which means it's going to be found throughout the cytoplasm of the cell, and you can see that represented over here on the right hand side. Now the cytoskeleton performs two functions. One of those is to give shape to the cell, very similar to what our skeleton does for our own bodies, but it can also be used to move various organelles or cell parts around inside of the cell. If a cell part is needed in another part of the cell, the cytoskeleton can be used to make sure that that organelle gets to where it needs to go. So one really important part of the cell that you really have to have a really good solid understanding about is the cell membrane. Now the reason this is important is because the cell membrane is going to regulate what can go into and what goes out of the cell. And it's going to be found in all cells. So it doesn't matter if it's a prokaryotic cell like bacteria, you're going to have a cell membrane. If it's going to be a eukaryotic cell, whether it's plant, animal, you will definitely have a cell membrane. Now, this cell membrane is going to be made up of two parts. It's going to be made up of a lipid bilayer. Now remember, lipids refer to fats, and we call it a bilayer, which means that there's going to be two layers of fats. And it's also going to be made up of protein. Down here towards the lower right hand side, you're going to see sort of a blown up view or a magnified view of that membrane. Now what you see in white here towards the top, that's your first fatty layer, and if you can barely see it right down here towards the bottom, that's going to be your second fatty layer. And this purple object that you see here, that's going to be what we consider a transport protein. We say transport because it can actually open up and it can close. So it gets to determine what's going to go into and what's going to go out of the cell. Now when you talk about substances in relation to the membrane, you will consider that substance either permeable or impermeable. A permeable substance can easily get past that membrane, so it can easily get inside the cell. If the substance cannot get inside of the cell, we consider that substance impermeable to that cell membrane. So again, this membrane is really important because it determines what can go in and what leaves the cell. Now up to this point, we've been looking primarily at an example of an animal cell. But what we need to do next is we need to make sure that we understand the differences between plant cells and animal cells. And one of the big differences is Plant cells have something called a cell wall. Now this cell wall, if you look on the right hand side, is indicated by this very dark green layer that you see right here. And right next to that green layer, you're going to see a very thin yellow layer, and that's going to be the cell membrane. So they lie really close to each other. Now the cell wall is super strong, and one of its primary functions is to provide support to the cell and the plant overall. Now that cell wall is made of cellulose, and if you remember back in 2.3 we had talked about the complex carb of cellulose, and that cellulose was only found in plants, and again that cellulose helps to provide the support that the plant needs to be able to stand upright. Now in addition to that support, the cell wall is also going to prevent the cell from rupturing, so it helps to protect the cell. Just in case the pressure within the cell would happen to increase, that cell wall is going to prevent it from exploding. So that cell wall is pretty important to the plant cell. So another big difference between a plant cell and an animal cell is that plant cells have something called a vacuole. Now animal cells have something similar, but we called them vesicles. Now the vesicles that we talked about in animal cells were associated with this structure down here called a Golgi apparatus. And remember, the Golgi apparatus secreted these vesicles so you could transport substances to different parts of the cell. Well, believe it or not, the plant cell also has vesicles, and you can kind of see them right here as these little blue dots. But they have a huge fluid-filled structure as well called a vacuole. Now this fluid-filled structure can contain water, it can contain salts, it can contain various proteins, and even carbs. But the big function of this vacuole is to provide pressure to the inside of the cell. Now, the best way to understand this is to think about a plant that has just wilted. In other words, you haven't watered the plant for a long period of time. If you don't water the plant, this vacuole actually will shrink. And when the vacuole shrinks, the pressure on the inside of the cell is going to decrease, which means the plant is not going to be able to remain upright. But once you water your plant, this area within the vacuole, the fluid within the vacuole is going to increase, which means you're going to have an increased amount of pressure on the inside of the cell. And when you increase that pressure, the plant now becomes upright once again. So again, this particular structure is really important when it comes down to not only storage, but allowing that plant to remain upright. So the final big difference between an animal and a plant cell is that plant cells have the presence of a chloroplast. Now the chloroplast is really important to the plant cell because it contains a green pigment called chlorophyll. Now that chlorophyll is important because that chlorophyll is going to be used to collect light energy or solar energy from the sun. And that energy itself is going to be transformed into a chemical energy. And that chemical energy becomes the food for the plant. Now this entire process of taking light energy and transforming it into chemical energy, as most of us know, is going to be called photosynthesis. Now over here on the right hand side you can see the chloroplast being represented right here. Now I had mentioned that I wanted you guys to make sure that you could recognize the difference between a mitochondrion and a chloroplast. So if you notice on the chloroplast, we have the chlorophyll, which is going to be found actually within these structures that are stacked on top of each other. So that's pretty unique to these organelles. This right here, this is a mitochondrion. And if you look really closely, you're going to notice the structures within the mitochondrion are in what we call a zigzag pattern. So you compare the mitochondrion to the chloroplast, you can definitely see a difference between the insides of both. Now here it's pretty evident between the two. You can see the difference because the chloroplast is much bigger in this cell compared to the mitochondrion. But there are some diagrams where they'll be very similar to each other, and you will have to look at the inside to be able to see the difference between the two. So again, zigzag pattern in the mitochondrion and a stacked pattern within the chloroplast. Alright, so that's going to wrap up this screencast for 7.2. As always, make sure that you've completed the screencast notes before you come to class.